want the people to appreciate, or the people who are working in the field appreciate, that the nomenclature they're using may not be appropriate, and that there is something they could use to make it appropriate and also understandable for everyone else.
Welcome to EyeCan, a podcast about ophthalmology through a uniquely Canadian lens with Hadi Saheb and myself, Heather O'Donnell. Season 5 of the EyeCan podcast is brought to you by Bayer Ophthalmology. We thank them for their support.
La saison 5 du balado EyeCan vous est offerte par Bayer. Nous les remercions pour leur soutien. We will be sharing our experiences as ophthalmologists today and tackle some of the challenges we face as healthcare providers. Professor Marion Munk is a trained MD, PhD, uveitis, and medical retina specialist. She is Chief Scientific Officer and Head of Research at Gutblick Group, Professor at Inselspital, University Hospital Bern, and Adjunct Professor at the Northwestern University in Chicago. She's Director of Ignos Consulting and Chief Medical Officer at Isarna Therapeutics. Dr. Munk is author of over 260 scientific articles. Her major research interests cover image processing, image analyses, artificial intelligence, macular diseases, and uveitis. She is research chair at the International Retinal Imaging Society, member of the editorial board of IOVS, ACTA, Ophthalmologica, Ocular Inflammation and Infection, Ophthalmologica, BMC Ophthalmology, and iNet, and is providing peer review for a long list of scientific journals within ophthalmology. Dr. Munk, we're so grateful to have you joining us today. We're excited to welcome you to the EyeCan podcast. So let's get straight into our chat.
Thank you so much for having me today. Thank you.
Great. So I wanted to start by having you share a little bit more about some of the nomenclature in uveitis and imaging. I think it's so important as we read the papers and listen to lectures to have a little bit more information about some of the nomenclature. So could you explain the importance of Delphi methodology in standardizing uveitis nomenclature and imaging?
Of course. Thank you for the question. So Delphi methodology and similar approaches, such as there's, for example, nominal group techniques or something else, they're particularly important in areas where practice patterns and diagnostic emphasis vary globally. And the Delphi method is a very structured communication techniques used to achieve consensus among a group of experts through multiple rounds of questionnaires. And believe me, there can be really multiple rounds of questionnaires. But typically what is very important, they're conducted anonymously. And after each round, a facilitator summarizes the responses and then provides feedback to the group, allowing the participants to revise their answers based on the aggregated input. And this iterative process continues until we have a predefined level of agreement. And then we can say we have reached consensus in a certain field or in a certain question. And in ophthalmology, particularly in complex areas like uveitis or imaging biomarkers, the Delphi method is really ideal for standardizing definitions where evidence may be still evolving. And expert judgment is really essential here. And it also reduces the bias from dominant voices because it's always kind of done by surveys. And so people can't overrule other people by kind of discussions. And it also encourages a broad input across specialities and geographies, and also leads to robust consensus-driven recommendations. For example, when we take our effort on OCTA standardization, we included biomedical engineers, retinal specialists, and also experts in retinal autonomy. And through the Delphi method, we can then distill a collective expertise into a harmonized definition, for example, also grading criteria and image interpretation frameworks. When we, for example, look at the consensus-based framework for OCTA in uveitis and retinal vascular diseases, we have to keep in mind that OCTA is an emerging imaging modality. So therefore, no guidelines exist what we can call wild field OCTA, for example, or what is significant amount of absence of OCTA signal due to retinal nonperfusion, for example. So what can be analyzed, and also the way we describe the images, it is very heterogeneous, and there are really no guidelines here. And this is very, very important to have guidelines so that we really make sure that when one person speaks of one thing, that the other person also knows what the other one is talking about. And when we look at this framework for the way we did it, so we initially sent out surveys to large uveitis and retina societies, including the Retina Society, the Euretina, the Japanese Retina Vitreous Society, and from the uveitis groups, the IUSG, the American Uveitis Society. And we asked them questions, and questions which did not reach consensus among these large societies were then presented to the expert groups to further agree on certain terms and in order to find valid recommendations. And so based, for example, on this work, we were able to agree that, for example, OCTA parameters like foveal avascular zones or area of absent OCTA signal consistent with retinal nonperfusions or presence of neovascularization visible in OCTA, all these kind of features should be really implemented in the new staging of diabetic retinopathy. We also agreed that, for example, diabetic macular ischemia can be diagnosed and can be classified and defined via OCTA, and that we can also differentiate between ischemic or non-ischemic RVO just based on the OCTA images alone. And we also agreed on definitions like that, for example, a meaningful change would be like a 30% or higher degree of absolute imaged area change in order really to see a response to a drug, for example. But there were also other aspects where we couldn't find consensus, and there, of course, we took it further, and we are still working on it.
So interesting. Historically, as you look at kind of the pyramid or the hierarchy of evidence, consensus has been, on the lower end, and it feels like, with the Delphi methodology and all the work you're doing, we're hopefully slowly allowing consensus that's brought forward through this process to have a little bit more weight in the hierarchy of evidence. So I think that's interesting. I'm curious to know if this process has brought forward any findings that totally surprised you. I think the beauty of consensus for somebody as involved as you in the space is that, likely there's not going to be any surprises. But were there any, as you went through this methodology, were there any surprises that came about through the process? Or do you feel like most of your conclusions were aligned with what you expected?
No, I think doing these kind of things, you learn a lot. You learn a lot about, the psychology of people. You also learn a lot about reaching consensus. And of course, there are several ways you can influence this. And as soon as you understand that, you really have to keep yourself in the background, not in order to kind of guide the expert group into some direction. I think this is very, very important. And I think what is also important is that as you mentioned, we need consensus and expert recommendation in fields and in areas where we do not have enough evidence to set certain algorithms here or certain way how to do things. But nonetheless, some level of evidence is needed, because if you have no evidence at all or like no data at all, it's really very hard to come to a consensus, because then it's purely opinion based, and you can come up with like everything. Yeah? So what is very important here is that you have to keep in mind that probably these kind of recommendations, they will change and they will evolve over time as more information and as more data are coming in. And it's always only a status quo. And regarding your question, yes, there were a couple of things which really surprised me. And for example, one thing was our effort in OCTA nomenclature. So as you know, in OCTA, when you describe a picture, when you look at the literature, it's very heterogeneous. So people use, flow void, flow attenuation, no flow, whatever. We did a literature review. We found over 200 descriptions for absent or missing OCTA signal consistent with retina nonperfusion. And it was in the survey as well as in the expert rounds, it was impossible to find a consensus here. And then we kind of regathered the groups and included more biomedical engineers and more people from basic research. And then actually we understood why this is the case. Because, for example, in OCTA, the way or the terms we're using, they are just not reflecting the physical principles of this device. Because when we look at OCT, the physical principles, what we see something which is hyperreflective, something which is hyperreflective. In fluorescein angiography, we see hyperfluorescence and hypofluorescence. However, in OCTA, we are not seeing flow. So the signal we are seeing there, it's not a quantitative measure, and flow indicates a quantitative measure. What we see is a binary thing, so whether flow is absent or present. So it's really like one signal of one subtracted OCTA where there was a change over time when the pictures were taken. And this is either it's there or it's not there, but it's not flow. And I think this kind of underlying principle has to be reflected in the wording we use in literature. And so it came that we really established a completely new framework how to describe these kind of OCTA images. And the initial effort in terms of agreeing whether we use flow attenuation or absence of flow or whatever, completely changed into the direction where we establish a complete new framework where we really say, okay, the only thing we can say what we see on OCTA is OCTA signal. So the only appropriate word here is signal, because this can be used for all the different machines. And from there, we can say there's absent OCTA signal or present OCTA signal. And then we can attribute that to the probable causes of it. So maybe, for example, like I said, OCTA signal due or absent OCTA signal due to nonperfusion. It takes a little bit of a while to kind of get this framework into your brain. But as soon as you start with it, it's as easy as saying no flow. And actually, this really reflects the physical principle. So this was really something which was surprising, that out of this effort, we established or we created a completely new framework, which is now kind of taken outside, and people use it more and more.
I'm listening to you, and there's a little bit of a smirk on my face because I'm wondering if now when you're at meetings and you hear people using maybe some older terms, are you tempted to kind of raise your hand or go up to the mic and suggest new terms, or have you maintained your ability to keep those comments in your brain?
That's a good question. I think 5 years ago, I would have jumped up and would have commented every single time. But now, when you get older, you're open to everything, and that it takes time. And there may be people who say, I don't do this. And I mean, you have to acknowledge and respect this as well. However, I just want to I want the people to appreciate, or the people who are working in the field appreciate, that the nomenclature they're using may not be appropriate, and that there is something they could use to make it appropriate and also understandable for everyone else.
Absolutely. In the glaucoma world, there are similar challenges in terms of surgical success and different kinds of surgeries and how we label success across studies and even just clinically. And so, I'm listening to you with interest, because I know that has been a challenge in the glaucoma space over the last years. So thank you for doing that.
Sounds interesting. We should talk more. Maybe we find that. Yes, I totally agree. So in terms of like you said, trabelectomy, for example, what can you call a successful trabelectomy? I can imagine this is yeah.
Yes, yeah. Absolutely. I mean, we made a lot of progress, but there's still enough heterogeneity that it becomes difficult to evaluate the literature. So I want to shift gears a little bit and talk a little bit more about injections. Injections are so omnipresent, and current retinal therapies, of course, heavily focus on these anti-VEGF treatments. So I'd like to hear your thoughts on what you feel the limitations of these therapies are, and how we can move forward.
Yeah, I mean, when it comes to injections, I think we have amazing anti-VEGF drugs now on the market. I mean, it's really, and I think we are complaining on a very, very high level. So I think we can do nowadays wasn't impossible like 20 years ago. But nonetheless, for example, when we think about, for example, neovascular AMD, we have to keep in mind that VEGF is the main driver for the acute exudative phase. Nonetheless, we have the underlying disease of AMD, so the degenerative, continuous degenerative and process is actually progressing. And this is not targeted by anti-VEGF drugs. They just make sure that the retina is dry. And these kind of secondary complication due to this continuous degenerative process, they are evolving nonetheless, no matter how effectively and how good you treat the patients with anti-VEGF medication. And this results then into functional vision loss, because the patients initially they gain vision, but then everyone knows over time they lose vision no matter what you do. And this is due to macular atrophy and fibrosis. So the point here is we have reached a very high, we have really reached a good treatment, and we have amazing drugs on the market, but still, like we see when we follow our patients, they lose vision. So I think there's definitely an unmet need here in order to develop drugs which are outside of this VEGF family and kind of really address unmet needs like atrophy or fibrosis. Or in retinal vascular diseases, for example, we know that the VEGF family is just one part of the driver of the disease pathology. I mean, we know that inflammatory cytokines, chemokines, they all play also a major role. And by only addressing one part of the picture won't solve the whole problem.
So on that topic, you're talking about the different mechanisms for vision loss in neovascular AMD and dry AMD as well. How has artificial intelligence helped you understand the different mechanisms of vision loss with AMD, and how has that had helped you in your future research project planning?
So first of all, I think I was an early adopter. So I think my first publication, which you can put in the field of machine learning, was 2010. So I think, I mean, like pros already worked with that a long time before, but nonetheless, I think for ophthalmology, it was quite early. And so I really tried to use it and like in all aspects, not only in my work area, but also, of course, in private. So I'm always astonished how much comes out. I always have the impression whenever I open my computer the next morning, something new has come up and something new you can try out, but whatever. In terms of AMD, for example, and in terms of this very classic way we use artificial intelligence nowadays is in terms of quantification of fluid, like automated detection of certain diseases. So I have found it. I use it in clinic, but I see there are so many limitations, starting not only about the process and the implementation, and then sometimes it's not working. For example, when we look at an OCT, so usually we judge whether the patient needs additional treatment or not, whether the macula is dry or not dry. However, when we have this quantification, sometimes we have a large discrepancy between the grading and the output from the AI. So for example, I see very often that I have a patient who have, 4 nanoliters of fluid. Nonetheless, I scroll through the OCT and for me, the OCT is dry. And so in order to implement this really in clinic and to have it as a tool to really use it in a fast way, we really need to understand thresholds, like what these numbers actually mean, and what like an increase of 10 nanoliters mean, what a presence of 4 nanoliters mean. And I think nowadays for us it's still more of something or a tool we play around with, or we still learn from it. And I think we will definitely need to implement all these AI tools into prospective trials and also have maybe exploratory endpoints based on these quantifications in order to really better understand, and that we can really use it wisely in clinics so that they really help us in decision making. This is one point. And another point I find AI very, very interesting is really understanding better the pathomechanisms of certain diseases. So, for example, I'm like you said, I'm in uveitis, and many autoimmune-driven diseases. Nobody understands what they're coming from, and I have the impressions I started being interested in white dot syndromes when I started, and I have the impression I have the same knowledge now after 20 years of being in this field. And with AI, it really helps you to better explore potential hypotheses we have. So, for example, like I said, I use AI also in terms of to explore whether there's a potential for molecular mimicry. So many autoimmune diseases, they are assumed to be triggered by molecular mimicry, meaning that you have maybe a pathogen, and the pathogen has a protein which looks similar and is folded similar than some proteins in the eye, and then the kind of then of course your body builds antibodies against this pathogen. But of course, because of the fact that this your protein in the eye looks so similar to the protein of the pathogen, it can happen that you have a cross-reactivity. This is molecular mimicry. And I started looking at this with different pathogens, and we really had like very, very interesting outcomes. And nowadays, and now having worked with that like maybe 3 to 4 years, I really think that something like molecular mimicry is one of the main drivers of many different diseases, because when you start seeing how many similarities there are between proteins in your body versus proteins of certain pathogens, for me, it's even a miracle that we do not have more of these autoimmune responses.
EyeCan wants to know what you think. Please send your comments on today's episode or any suggestions you may have for topics or features to communications@cos-sco.ca, and we'll try to incorporate them into future episodes.
EyeCan veut savoir ce que vous en pensez. Veuillez envoyer vos commentaires sur le balado d'aujourd'hui ou vos recommandations de sujets à communication@cos-sco.ca et nous essaierons de les intégrer dans les prochains épisodes.
Hi, I'm Brian Balios, and I listen to the EyeCan podcast.
Can you tell us more about your research on SARS-CoV-2 and tuberculosis and how this is impacting our patients in ophthalmology?
Yeah, sure. So I already started. So this was some work we did in terms of understanding potential molecular mimicry due to different pathogens. And we started doing this during the COVID phase. So of course, it was logical to start with SARS-CoV-2, because also there were so many discussions on, whether you can have autoimmune responses due to the vaccines, autoimmune responses due to COVID infection. And of course, we thought, okay, we may see more vein occlusions in patients, or some like AMPI-like pictures are really triggered by COVID. And what we did there, we really looked at the proteins of SARS-CoV-2 and compared them with the proteins of photoreceptors and RPE and ganglion cell layers. And what we found interestingly was there were no similarity in terms of photoreceptors, but there were some minor similarities in terms of the 3D structure, in terms of the RPE. And some of these proteins also had the ability to bind to immune cells. And then they also had the ability to cause an immune response. Because the question is always, so first you ask yourself, okay, is there a protein similarity in the structure? Then you go further, then you look whether the protein is folded in a similar way. If you find that the protein is folded in a similar way, you go further and look whether the immune cells actually can recognize these proteins, because who cares about a protein if the immune cells can't recognize it? And the third part is, even if the immune cells recognize it and kind of binds it, still it can happen that there is no immune response. So you really have to check all these 4 pathways, and you can do this all with AI and bioinformatics. And here we found some similarities and also some similarities where some RPE proteins could induce this kind of autoimmune response. But they were really only little, and I think there were only 3. But however, when we looked at retinal endothelial cells, we found so many different retinal endothelial cells protein, which really showed significant similarities between SARS-CoV-2. And also many of them had very strong ability to cause an immune response. And for me, having these results and then looking into clinic and seeing like the higher prevalence rate of retinal vascular events, for example, and maybe also some cases of MP-like pictures, and also like the higher prevalence rate really in retinal vascular events here. So this really makes sense. And I think this is really something which is real. And the higher events of these retinal vascular diseases probably are not only coming from this higher hypercoagulable state and the higher inflammatory state by the disease itself, but rather really also by this autoimmune cross-reactivity of the endothelial cells in our body, which then, of course, reacted and caused an immune-mediated shutdown or kind of occlusion.
So interesting. I'm curious if you found anything similar with vaccines, or if this was unique to the actual viral infection.
No, this is actually not only the viral infection, because the protein which is able to cause it is also included in the vaccine. So from this theoretical work, a vaccine has the same ability to cause a molecular mimicry or an autoimmune response than the infection itself.
Moving on to giving you a crystal ball and asking you to think a little bit about the future, since you're so involved in this space. I'm wondering if you can predict what might be some exciting opportunities for innovation in ophthalmology in the next decade. What's your best guess?
It's very hard. It's very hard to predict the future. I think, but I don't even know where to start. So I think, first of all, disease-modifying drugs. And I mean, really disease-modifying drugs we know from, for example, rheumatology or also multiple sclerosis, where we can really preserve vision over longer time and kind of change the trajectory of the disease itself. I really see this with maybe new drugs with new mode of action and with new endpoints evolving, that this should be possible in the future. Then, of course, also the implementation of smart imaging and remote monitoring of our patients, no matter whether glaucoma or patients with retinal or macular diseases. And then, of course, the integration of systemic and ocular biomarkers for a more personalized eye care. So if you ask me, I would really envision like a hospital or a center where every patient is initially kind of evaluated and assessed by different AI tools. And based on this, then as a second step is seen by the doctor.
Fantastic. I'd like you to humor me through what I'll call a lightning round, and just some short questions that are meant to be answered in a few words. Some are professional, some are personal, and hopefully it's fun. So I want you to start, if you can tell me what one imaging tool is that you couldn't live without in your practice.
OCT.
Of course, I figured.
Yeah.
2, do you prefer seeing medical retina patients or uveitis patients?
Oh my God, come on. This is so hard. I can't answer this question. So it really depends on my day. So if I have slept enough, if I'm like looking forward to the day, I prefer uveitis cases because they're more challenging. It's always like a riddle. I always feel like Dr. House when I see an uveitis case, especially when you have referral for a second opinion, nobody knows what it is. It's just very, very, it's really exciting. On the other hand, if I have a lot to do and I'm very tired, I really prefer medical retina patients because they're just much faster. You have an algorithm in your head where you do not have to think so much, and it's just much faster to go through. So I would say a mixture of both of them make a perfect clinic day.
Sounds great. Next, your favorite AI tool in your personal life.
This sounds boring, but I think it's still ChatGPT. It can do everything. I mean, I use it for example, for nomenclature group to find out what the best possible time for all time zones is to have the call. I use it for like fast assessments of anything. So I think it's still ChatGPT. So I think if you would ask me whether I rather need ChatGPT or a secretary, I would say I go for ChatGPT if I have to choose one. Yeah.
Wow. Well, my next question might have the same answer, but so the first was your favorite AI tool in your personal life, and the next was your favorite AI tool in your professional life.
In a professional life, I think it's also ChatGPT, but there are a lot of so for example, there is Elicit. I just recently found it out. It's really a great tool for systemic reviews. And the prior ones I tried out, they're not so good, but Elicit is really good. So if you want to get an overview on a certain topic, it's amazing. What else? There are so many tools. It's just of course analysis tools like a Retinal for automated quantifications. I think I can't imagine my personal as well as my private days without the AI tools anymore. Yeah.
Okay. And my final question, this lightning round is, do you prefer Swiss chocolate or Dubai chocolate?
Oh, this is easy. Swiss chocolate.
Aha, great, great. Well, thank you. So as we wrap up our chat today, we want to hear a little bit more about what you do in your non-work hours. You've alluded to some of it already, but any other hobbies or interests that you're willing to share with us today?
Of course. So I think this will be also in line with a lot of Canadians. So I love nature. So I spend every single time possible in nature. So when I write papers, when I work outside, when I have calls, so usually I'm sitting outside and try to do this from outside, and all kinds of sports outside. This is what I do. This is one thing. So I really dream of a wooden cabin in the mountains with like near mountain spring and perfect vantage point for my telescopes. So this is like my dream. And otherwise, reading, so fiction and nonfiction. I know it sounds very boring, but I really enjoy that.
Sounds like you need to move to Canada. We have tons of opportunity to meet your interests, so that's great.
My brother already lives there, so he tries to convince me. So I might.
Great. Well, hopefully one day. Marion, this has been such a great conversation. Your interests are so broad and so relevant, and you've done such great work and had such an impact on our field. So thank you for sharing some of the highlights of your interests with us today. And we look forward to hearing more about what you do in the future. Thank you again.
Thank you so much.
Thank you for joining us for this episode. We look forward to bringing you more engaging content over the coming months. EyeCan wants to know what you think. Please send your comments on today's episode or any suggestions you may have for topics or features to communications@cos-sco.ca, and we'll try to incorporate them into future episodes.
EyeCan veut savoir ce que vous en pensez. Veuillez envoyer vos commentaires sur le balado d'aujourd'hui ou vos recommandations de sujets à communication@cos-sco.ca et nous essaierons de les intégrer dans les prochains épisodes.
Season 5 of the EyeCan podcast is brought to you by Bayer Ophthalmology. We thank them for their support.
La saison 5 du balado EyeCan vous est offerte par Bayer. Nous les remercions pour leur soutien.
Thank you from the Canadian Ophthalmological Society. EyeCan is available wherever you listen to your podcasts and, of course, through the COS Practice Resource Center.
EyeCan est disponible partout où vous écoutez vos balados et bien sûr au Centre de ressources pour la pratique de la SCO.
EyeCan is written and directed by Kim Teitler and produced by John Allaire from Allaire Strategic Works.
EyeCan est écrit et réalisé par Kim Teitler et produit par John Allaire d'Allaire Strategic Works. Some episodes this season will include relevant links and show notes, so be sure to check episode descriptions to learn more. Certains épisodes de cette saison incluront des liens pertinents dans les notes d'émission, alors n'oubliez pas de consulter les descriptions des épisodes pour en savoir plus.
If any of our listeners have topic ideas to submit, please email them to communications@cos-sco.ca, and we will review for consideration.